Hello friends, so welcome to the Agriculture channel. So today we are discussing about the different mechanism behind male sterility and what is the role of male sterility. We will see majorly this male sterility will be used for hybrid seed production. So what do you mean by male sterility? So male sterility means non-functional pollen. Male sterility means sterile with respect to the male but fertile with respect to the female. So the plant which is male sterile in nature, so what we are having non-functional pollen but female gamete function normally. So let us understand what is the role of male sterility and how it will help in the hybrid seed production. Let us come to this part. There is one is plant 1 and another is plant 2. So in this case, suppose I am taking rice. Suppose it is a rice plant. Okay. So rice is a which type of crop? It is a self-pollinating crop. Which type of crop? It is a self-pollinating crop. So normally what happen in case of rice, the self-pollination means so whatever the pollens will be released from the anther, this pollen will fall over the stigma of the same flower. That means the pollen which will be released from the same flower or from the anther which is present in the same flower will fall over the stigma of the same flower. This is called as the self-pollination. So what is happening? Whenever we are going for hybrid seed production, we are taking two types of parent 1 and another is parent 2. So what we need the suppose parent 1 is female 1, parent 2 is male 1. So what we need the pollen of the parent 2 will fall over the parent 1 and we will having the F1 hybrid seed. Okay, so this is a whenever we are going for hybrid seed production, what we need cross pollination. What we need cross pollination. Cross pollination means the pollen from one parent will fall over the stigma of the another parent, that is female parent. So this is called as the cross pollination. So that means suppose I want to produce hybrid rice seed. So that means I want to follow which pollination? Cross pollination. So because in case of rice plant, rice is a self-pollinated crop. So as for hybrid seed production, what we need? Cross pollination. But in case of rice, self-pollination is its natural pollination behavior. So in this case, what we are going for? We are having different methods. So suppose what I will do, suppose there is a technique, that is suppose I can remove this anther. Suppose if I will remove this anther, then what will happen? So from another, the pollen from another plant will fall over the stigma here. Okay, so now what is this is called the cross pollination, and the seed produced in this plant is called as the hybrid seed. Okay, so this is called as emasculation. Emasculation means removal of the anther before dehiscence of the pollen. So before release of the pollen from the anther, what we are going for? We will remove this anther so that no pollen will be released and there will be no self-pollination. Okay, so it will automatically allow the pollen from the other plant fall over this stigma pores. So by emasculation technique, we can also go for the which there is hybrid seed production. But imagine, so is it easy to remove all the anthers of the flower present in the plant? No, it is not possible to remove all the anthers. So that is why what we are going for, we should go for male sterility. Which procedure we should follow? Which procedure we should follow? Male sterility. So male sterility, our focus should be whenever we are going for preparation of the male sterile plant, our focus must be though the anthers are present, in the flower, but what is the pollen which will be produced? This pollen should be non-functional. This pollen, though this pollen will be released and it will be fall over the stigma, but this pollen is which nature? The nature of the pollen is non-functional. Non-functional means Now this pollen cannot go to the ovary and cannot fertilize the ovule. So this is non-functional. The pollen is not functional. That is why this plant is called as the male sterile plant. This plant is called as the male sterile. That means the male sterile means the pollen which is produced in this plant is non-functional, or either the pollen is produced non-functional, or maybe the anther there is a production of a hard seed coat, so that there will be no dehiscence will be occurred. So non-dehiscence anther will be there, so no pollen grain will be formed. And if pollen grain will be formed, then the pollens are non-functional in nature. Hence, now the stigma or the ovule is fertilized by the pollens which will be released from the other plant, okay, from the plant 1. So suppose this plant 1 is male fertile. Male fertile means the pollen released here, the pollens released here, these are functional in nature. Functional nature means whenever it will fall over the stigma, it will fertilize the ovule and a seed will be produced. In which part? In which plant? Plant number 2. Hence the plant number 2 is called the male sterile or female plant. This acts as a female plant because now it will produce the seed, and the seed is produced by the pollen which will coming from the which plant? There is a male plant, male fertile plant. Okay, so this is the concept behind the male sterility. Hence the male sterile plants which will act as a female plant, it will be used for hybrid seed production. This is the concept behind male sterility. Okay, then coming to male sterility, it is divided into 3 groups. One is genetic male sterility, then cytoplasmic male sterility, then cytoplasmic genetic male sterility. 3-1, genetic male sterility, cytoplasmic male sterility, and another is cytoplasmic genetic male sterility. So before coming to these 3 groups, let us understand one concept. Suppose this is your plant cell. So you are having plant cell. So in this plant cell, you are having the nucleus. So this is your nucleus, this is your vacuole, and another chloros in case of in the cytoplasm. So one is nucleus and other rest part is called as the cytoplasmic organelles. So in the cytoplasm, you are having the chloroplast, and then you are having the mitochondria. And you know that whenever we are going for DNA, there is the gene. Gene means the DNA. In case of plant, the DNA is present both in nucleus as well as in the cytoplasm. So when the DNA or the gene is present in the nucleus, it is called, if suppose one male sterility gene, which gene? Male sterility gene. If the male sterility gene is present in the nucleus, then it is called as the genetic male sterility. So here in case of genetic male sterility, the gene is present in which region? In the nucleus region of the plant. Okay. Then next coming to, there are 2 organelles. Which organelles? Cytoplasmic organelles. One is chloroplast, which is present in the cytoplasm, and another is mitochondria. It is also present in the which part? In the part of cytoplasm. So now the thing is that this DNA, if this DNA present either in the chloroplast or in the mitochondria, this is called as the cytoplasmic male sterility. So if your gene is present either in the cytoplasm means either in chloroplast or in mitochondria, then this type of male sterility is called as cytoplasmic male sterility. Okay, but again you can see, if the gene is present both in cytoplasm as well as in the gene, suppose in a plant the male sterility gene present both in cytoplasmic organelles, either chloroplast, mitochondria, we will see whenever we will cover the cytoplasmic male sterility, we will see. Okay, or as well as in the nucleus, so it is called as the cytoplasmic genetic male sterility. So here gene is present both in the nucleus as well as in the cytoplasmic organelle, either in a chloroplast or in the mitochondria. Okay, so I hope you have understood the concept up to this point. So let us remove this thing. So okay, so now we will cover this thing one by one. So now let us first understand what do you mean by genetic mentality. I have already told genetic mentality means what? Now the gene will be present in which region? In the nucleus region. Okay. So now the genetic mentality which is present in the this genetic mentality, the gene, which gene? Recessive nuclear gene. Which gene? Nuclear gene. That is the genetic mentality are mostly which nature? Are recessive in nature, which is the nature is recessive. And you know, whenever we are going for character expression, there are two types of things. One is either recessive gene, or we are having the dominant gene, Suppose I am saying male sterile. Male sterile, which gene? So recessive gene. Recessive gene means when both of the alleles are recessive, then it will show the male sterile. Remember the Mendelian characters. So for recessive, in case of dwarf gene, if both alleles are small t, then the plant becomes dwarf. Okay, so recessive gene will be expressed. The recessive character, which one is the recessive character? The dwarf height, this is the recessive character. And the dwarf character will be expressed when both of the alleles are recessive allele. But dominant gene will be expressed in both, either in heterozygote condition as well as in the homozygote condition. So dominant gene means what? This becomes male fertile. Both of these becomes male fertile because here capital MS means male fertile. Because the male sterility is controlled by which gene? Male sterility is controlled by small ms gene. Which gene? Small ms gene. So as the male sterility is controlled by small ms gene, there is a recessive gene. So according to the Mendelian principle, the recessive character will be expressed when both of the alleles are recessive. So like in small t, the plant is dwarf. But in case of capital T, small t, heterozygote condition, the plant is tall. And capital T, the plant is tall. So whenever we are having dominant gene, the dominant gene expressed in both heterozygote condition. Heterozygote means one allele is dominant, another allele is recessive. So this is your heterozygote condition. This one is your heterozygote condition. This one is your homozygous condition. So dominant gene is expressed both in heterozygote as well as homozygous condition, but recessive gene is expressed in which condition? Always in homozygous condition. This is which condition? Homozygous condition. Which condition? Homozygous condition. Okay. So now I think you have understood this part, genetic male sterility. Okay. So let us come to this part. So genetic male sterility, I have told you, mainly genetic male sterility is controlled by which gene? Is controlled by recessive gene. So the plant which will become male sterile means now this plant act as a whole female, 100% female, means this plant will produce the seed. This is your concept. Okay. So as the genetic male sterility is controlled by which gene? Recessive gene. Hence the plant becomes male sterile when both of the alleles are recessive or homozygous condition. Okay. And as I have told you, this gene is present with respect to genetic male sterility. This gene, recessive gene, should be present in which part? In the part of nucleus. In the part of nucleus. Okay. Okay. So now let us. This is, suppose this is, we are going for maintenance of male sterility. So how we will maintain the male sterile line? Suppose in this case we are having the A line. A line means male sterile. I have told you. Male sterile means we are going for genetic male sterility. So the gene will be present in the nucleus. So this is your nucleus. This is your nucleus. So inside the nucleus, both of the alleles are recessive. Then only it becomes male sterile. So male sterile means it acts as a female. Okay. Another line, B line. B line called as the maintainer line. Which line? Maintainer line. What it maintains? It maintains which line? It maintains the A line or male sterile line. This B line, what is the use of B line? The use of B line is to maintain the A line or to maintain the which line? Male sterile line. Because once you are using this male sterile or female line for seed production, from where I will get the seed of this A line? So that is why we have to go for maintenance purpose for A line. Suppose we are having 100 number of A line, and all the A line means the male sterile line, and all 100 number of female plants are used for seed production. Which production? Hybrid seed production. So if I will use all the 100 male sterile plants for seed production, then for next year, suppose this is 2026. Suppose in 2026 I am having 100 number of male sterile plants, and I am using this male sterile or female plant for hybrid seed production. So in 2027, from where I will get the male sterile line? That is why we have to maintain the stock of the A line. So how you will maintain? Just cross between two lines, A line and B line. What is the use of B line? You can see here in the nucleus. What is the genotype? This is heterozygous condition. Which condition? Heterozygous. Heterozygous means one is dominant, another is recessive. So now cross these two line, L and B line. What I am getting? So 50% I will get what? This is called as small ms and small ms come together to produce male sterile line. Why male sterile? Because both of the genes, both of the alleles present within the gene, and the gene is present in the nucleus, both are recessive. Hence it acts as a male sterile. Again, what will happen? The capital MS and here small ms come together to produce male fertile. Why it is male fertile? Because one allele is dominant. I have told you. So whenever dominant, one of the allele will be dominant, means the dominant character will be produced. So in heterozygous condition, so it is a heterozygous condition. Two different types of alleles are there. It is a homozygous condition. So in heterozygous condition, what will be produced? Male fertile line will be produced. Male fertile means this act as a male parent and this act as a female parent. So I will cross one male, one female, another which B line, which acts as a maternal line. This is which type? It is heterozygous type. This is homozygous. So I am having 50% female, 50% male. Okay. So now from this I am having 50% female parent. So that is why. So by crossing the B line, what I am getting? I am getting female line. So that is why the B line, which is called as a maintainer line, what is the use? What is the purpose? To maintain the male sterile lines. Okay. Then next come to seed production. So utilization hybrid program or hybridization program or hybrid seed production. So in case of hybrid seed production, we need what? We need only hybrid seed. So for hybrid seed, so one act as a female, that means other part. So I have told you na, in hybrid seed production, one act as a female parent, another act as a male parent. So from the male parent, the pollen will fall over the female parent, and now the progeny act as a F1 hybrid. So in hybridization program, so I am having the female parent. there is male sterile line, which is characterized by recessive gene small ms. Okay, so it acts as a female. Okay, in the next line I will cross this with one, with male fertile line. Male fertile means here 100% male lines will be there, that is homozygous but dominant gene. So it's because all are dominant gene what I am having. So you can see all the plant, whatever the F1 hybrid I will get, whatever the F1 hybrid I will get, all the plants of F1 hybrid are heterozygotes. These are heterozygotes. And if I'll ask you whether this is male sterile or male fertile, so you will see whether it is homozygous or heterozygous. So this is heterozygous in nature. One is dominant, another is recessive. So as it is heterozygous, which allele will be expressed? The dominant allele will be expressed. That is dominant will be male fertile. So that is why 100% F1 what I will get, all are male fertile. Okay, so this is the concept behind the genetic male sterility. Okay, so I think you have understood this part. So now again this genetic male sterility based upon the environmental sensitivity, it is again divided into two categories. One is environmental sensitive, another is environmental insensitive. So environmental insensitive means what? Whatever the genes I am having, that is male sterile gene, this one, this is not affected by the environment, which is called as the environment insensitive gene. Suppose in some of the cases this gene, which gene? Recessive male sterile gene MS, it is environmental sensitive. So if it is environmental sensitive, I am having two things. Either one is temperature, another is photoperiod. Another is photoperiod. So temperature means what? So in this case, temperature means if your temperature is more than 23.3 degrees centigrade, if your temperature is more than 23.3 degrees centigrade, now my plants will act as a male sterile. So means high temperature leads to male sterility. Example, rice temperature sensitive genetic male sterility line, there is PAI65. So coming to photoperiod sensitive, photoperiod means, as you know that if in long duration condition, also long day condition, if the day length, photoperiod means day and night, photoperiod means day and night length. So in long day condition, or if your day length will becomes greater than 13 hour 45 minutes and the temperature should be 23 to 29 degrees centigrade, my plants will become male sterile. Or we can say the small ms gene which is present in the nucleus becomes active or homozygous condition. So this is called as the environmental sensitive condition. Okay, so this is the two group, environmental sensitive and this is insensitive, temperature, and another is PGMS. Just you have to remember the temperature and the day length. Okay, so we have seen another type of genetic male sterility which is called as the transgenic genetic male sterility, called as transgenic genetic male sterility. So what is happening? So in this transgenic male, you will see here, so let us remove this one. So what we are going for? So transgenic genetic male sterility. So in this case what we are going for? So suppose this is your plant and this is your nucleus, and in this nucleus a DNA is present and gene is present here. Okay, transgenic means what? So, suppose I am having one bacteria. Which bacteria? I am taking Bacillus amyloliquefaciens. Bacillus amyloliquefaciens. So this Bacillus amyloliquefaciens, it has a gene. Which gene? This is called as which gene? Barnage bar gene. Which gene? Barnage bar gene. This Barnage bar gene, what is the use? this Barnage bar gene is used for the development of the male sterility. How? You can see this is called as the anther. This is called as the anther. So in this anther, this is called as the tapetum cell. Which cell? Tapetum cell. Which cell? Tapetum cell. So what is the role of tapetum cell? Inside the anther, which one will be produced? Your pollen grains will be produced. So what is the role of the tapetum cell? So this is your tapetum wall or tapetum cell. The role of tapetum cell is to provide nourishment, to provide a nutrient to the pollen grain. Remember, the role of the tapetum cell is to provide nutrient to the pollen grains. But what is the role of Barnage bar gene? So once I will transport this Barnage bar gene from this bacteria to your plant. Suppose I will transport this gene. Suppose now I will transport this gene to your, this is your plant. Suppose I will transport this which gene, Barnage bar gene, to your plant. So what will happen? Now this in your plant itself, which gene is present? Barnage bar gene is present. So now the plant becomes transgenic because you are transferring this gene from bacteria to your plant nucleus gene. So now what is the role of Barnage bar gene? This Barnage bar gene which will produce one RNase enzyme which will break down the RNA and it will affect the function of the tapetum cell. It will stop. This Barnage bar gene will stop the function of the tapetum cell. So if the tapetum cell function will be stopped, that means there will be no nutrient will be provided to the pollen grains. So now your pollen grains becomes deactivated, or now the pollen grains will the pollens becomes non-functional. Okay. So now Barnage bar gene helps in, if we'll say in a simple case, this Barnage bar gene helps in development of non-functional pollen. That means this Barnage bar gene helps in development of the male sterile line. Simple. In another bacteria, in the same bacteria, another gene is also present, which is called as the Barster gene. Which gene? Barster gene. This Barster gene will stop or it will prevent the function of the Barnage bar gene. So if in one plant Barster gene will be present, then it will prevent the role of Barnage bar gene, and it will now if the plant present in the if the plant contains Barster gene, now the plant becomes male fertile. Remember? So let us, I am saying repeatedly, Barnage bar gene, which is come from which bacteria? Bacillus ameloleucophaciens. If this Barnage bar gene will come to your plant, there is nuclear gene, or which becomes the transgenic gene. So now what is the role of Barnage bar? The role of Barnage bar is to prevent the function of the tapetum cell, thereby it will prevent the supply of nutrient to the pollen grain, to the pollen. So now your pollen becomes non-functional. So your plant becomes male sterile. This is because of Barnage bar gene. But if that plant contains another gene which is called as a Barster gene, this Barster gene also comes from this bacteria. Suppose this Barster gene also present within this plant. So now this Barster gene, will stop the function of the Barnage bar gene. So now this process will be stopped. So now your plant becomes male fertile. Okay. So now here we are saying, suppose one parent I am taking, that is Barnage bar gene. Suppose one plant that is Barnage bar gene. So this is two allele. This is normal allele. Another allele that is Barnage bar gene. So if Barnage bar gene is present within a gene, if Barnage bar is present, then I have told you the plant becomes male sterile. In normal case, there is no Barnage bar gene, so normally your plant becomes male fertile. And here we are having those plants which are having Barnes bar gene, they are phosphinothricin resistant. That means those plants contain the Barnes bar gene, these are phosphinothricin resistant. And again I have told you Barnes bar containing plant are male sterile. Okay, the first concept. So if I will cross between these two plants, one is containing Barnes bar gene, another is male fertile, which is normal plant. So I am having two types of plants. One plant contains Barnes bar gene, another I am having the normal plant. So if this plant contains Barnes bar gene, so now my plant becomes male sterile. How much percent? 50%. Okay. So rest 50% I am having male fertile, because you are saying this. So here having from this, suppose this is female parent, this is your male parent. Okay. Male sterile means female. So from Barnes bar from one parent and normal gene from other parent, 50%. So normal gene from one parent and normal gene from another parent, this is called as 50% male fertile. So now if I will apply phosphinothricin to this population, then what will happen? The plants which are male sterile, which are containing the Barnes bar gene, they will survive. But the plants which is Barnes bar gene is absent, they will be killed. So now these Barnes bar plants are survived. So now what will happen? If I want to produce more amount of male fertile lines, then what I need? I need Baxter gene. So whatever the progeny I will found here, so I will cross with another parent that is containing Baxter and Baxter gene. So now see, this is Barnes bar gene and Baxter gene will come. This becomes male fertile. Why male fertile? Because if Baxter will be produced, but if Baxter gene is present, it will prevent the function of Barnes bar gene. So now here plants becomes male fertile. So here normal gene will come and here Baxter gene will come. Again you are having the male fertile. So here 100% plants I am getting all are male fertiles. So this is a type of called as the transgenic genetic male sterility. So I hope you have understood which portion genetic male sterility. And in the next portion or the next lecture we will go for cytoplasmic male sterility and cytoplasmic genetic male sterility. So okay, thank you so much.